activator-mediator interaction. First, I would like to briefly introduce the background information of this paper. First is the azole antifungal agents, and they can prevent the fungi mainly by the prevention of ergosterol synthesis. And ergosterol is a critical compound of fungal plasma membrane, and they will inhibit the 14-alpha sterol demethylate. And the most widely used antifungal agents nowadays are fluconazole and ketoconazole. So in this paper, they will focus on these two azole antifungal agents. And the resistance of the antifungal agents was first documented in 1993. So it has been more than 2 decades. Then is the fungi. The paper focused on Candida glabrata and Saccharomyces cerevisiae. They were originally thought to be non-pathological organisms, and they belong to PDR, pleiotropic drug-resistant subfamily, because they contain a PDR gene, and that is associated with multi-drug resistance to azole. So the prevalence of these two fungi is due to the multi-drug resistance of azole, and the treatment of azole is widely used now, so the prevalence of them will be very critical and cause many obstacles on clinical therapy. And they can gain their drug resistance to azole simply by a gain-of-function point mutation. Next is a brief concept of the mechanism the paper talks about. It is the PDR1 gene, and the drug efflux genes will be driven by the expression of PDR1 gene, and the PDR1P is the activator of PDR1 gene. In the presence of the toxic compounds such as ketoconazole or fluconazole, the activator will be activated, and it will recruit the mediator through the protein-protein interaction of the activator with the mediator KIX domain. So the drug efflux gene will be upregulated, and the function of it is the drug efflux pump. It will transport the toxic compounds such as azoles out of the cell, so making the fungi free of the toxic compound. So they will gain the drug resistance to azole. And in this paper, they focus on the interaction between the activator PDR1P and the mediator KIX domain. So they try to inhibit the protein-protein interaction and hoping to reduce the gene expression and the function of drug efflux pump. Then is the first part of the result. First, they try to discover the inhibitor, the most potential inhibitor to inhibit the activator and the mediator, so that the PDR1 gene that is responsible for the drug resistance will not be expressed. First is the screening process. They use in vitro study to narrow down the candidates, and then they use in vivo Saccharomyces cerevisiae viability screening. And this is the most potential candidate, the IKIX1. The IKIX1 can reduce the Saccharomyces cerevisiae viability in a most efficient way. And this is the chemical structure for IKIX1. Then they will do some more characterization of IKIX1. This is the dissociation constant of the activator and the mediator. The constant is about 0.32 micromolar. Then they use the IKIX1 to compete with the activator's interaction with the mediator. The half maximum inhibitory concentration is about 18 micromolar. Then the second figure, they want to identify the interaction interface of the inhibitor IKIX1 with the activator and the activator with the mediator. Then is the mediator KIX domain in Candida glabrata and Saccharomyces cerevisiae. They have 51% sequence identity. and their structural prediction, their structural alignment is very similar. They contain 3 major helix domains. Then they use the chemical shift perturbation methods to identify the interaction interface of the activator to the mediator and the inhibitor IKX1 to the mediator. And they show that the similar interface is the red part here and is the blue part here. They majorly targeted the hydrophobic group. Then they use the method of docking to predict the IKX1's binding site with the mediator. And the binding site shown is here. And the binding residues are mainly hydrophobic. That is consistent with the previous study. And the location of the predicted binding site is very similar to the previous, is also very similar to the previous study that is in the hydrophobic major group. And the interaction suggested that there are also hydrogen bound and hydrophobic interactions. And it demonstrates that the interaction between the IKX1 and the mediator is very strong. So the series of experiments, it shows that the inhibitor IKX1 can compete with the activator to interact with the mediator KIX domain. Then they examine whether the IKX1 will recruit the mediator and whether it will regulate the PDR gene that is associated with drug resistance. This is they use the luciferase gene driven by the PDRE gene. And with the treatment of ketoconazole, the luciferase activity detected will increase. And this is very reasonable because in the presence of ketoconazole, the PDRE gene that is associated with drug resistance will be increased. So the luciferase activity will increase. But the luciferase activity will be reduced with the addition of IKX1 and in a concentration-dependent manner. Then they use the chromatin immunoprecipitation assays to analyze whether the mediator will be recruited by the IKX1. Also, it was the recruitment of IKX1 will rise with the treatment of ketoconazole, but will be decreased with the co-treatment of 20 micromolar of IKX1. This is the RNA level tested. There is also an azo-induced expression, and it will be abrogated by the treatment of IKX1. So it shows that IKX1 treatment can abrogate the mediator recruitment to the upstream activated sequence. Then they try to show that the PDR1 gene expression, the CDR1 and the CDR2, the expression and the function of IKX1. The expression of the PDR1 regulated gene will also be increased when the addition of ketoconazole and will be reduced by the addition of IKX1 and also in a concentration dependent manner. And on the both PDR regulated gene, they shows a similar expression. So due to this experiment, it suggests that the addition of azo will induce the PDR1 dependent drug efflux pathway, but the entire pathway can be blocked by the addition of IKX1. Then they isolate the drug resistant strains of Candida cabretta, and there are 5 different strains, and their expression and they have drug resistance mainly due to they have gain of function point mutation at different sites, and their expression of the PDR1 dependent gene will be reduced by the addition of IKX1. And after a series of molecular level screening, they will identify the function of drug efflux pump. So they examine the drug efflux rate. And they use the PDR1 is the wild type Candida cabretta. And these two are the gain of function mutant strains. So the efflux rate of the drug efflux pump will decrease by the addition of the IKIX1, and these are the blue bars. So it suggests that the addition of IKIX1 can actually interrupt the relationship between activator and mediator, and it can be used to decrease the expression of the gene and abrogate the recruitment of mediator, and also decrease the function of the JOG efflux pump. Then is the in vivo assays of IKIX1. Figure A is a diagram of the point mutation site, and in different sites of point mutation, the Candida glabrata mutant can gain the azole resistance. So in the figure, this one, only the wild type strain, the first strain, the wild type strain will be sensitive to ketoconazole and fluconazole. All these 6 mutant strains are not sensitive to the addition of azole drugs at all. With the addition of IKIX1 in different concentration, not only the wild type strain will be sensitive to fluconazole and ketoconazole co-treatment with IKIX1. The mutant strain that originally have the azole resistance will be influenced, and the growth of this Candida glabrata will be reduced. So it suggests that IKIX1 can restore the azole sensitivity of these mutant strains. Then is an in vivo pathogenicity model, and they examine the wild type Candida glabrata and the JOG resistant point mutation strains of Candida glabrata. In the wild type strain, the addition of fluconazole alone is the blue line, is able to significantly increase the survival rate. However, the co-treatment of fluconazole and IKIX1 do not have obvious difference. However, in the strains of point mutation gain of function strains, the addition of fluconazole alone does not make significant difference. It is the co-treatment of fluconazole and IKIX1 that can prolong the survival rate of the survival rate. Then they use the mouse model, and they use tail vein injection of Candida glabrata to the mouse, and they then detected the fungal burden in both kidney and spleen. And it can be separated into 2 groups, wild type and the mutant. In the wild type, it shows very similar results that the fluconazole alone is able to decrease the fungal burden in both kidney and spleen. However, in the wild type, the treatment of only fluconazole does not have significant decrease in fungal burden. And the fluconazole treatment with IKIX1 is able to significantly decrease the fungal burden in both kidneys and spleen. So they use different in vivo model to suggest that the function of the IKIX1. So I would like to summarize with this mechanism again. They identified the PD-L1 gene is associated with the JOG efflux gene, and the activator and mediator relationship, the protein-protein interaction between them is very critical for the emergence of JOG resistance. So they screen for the inhibitor IKIX1 to block the protein-protein interaction and hope to decrease the function of JOG resistance. So to conclude the entire paper, the emergence of azole-resistant Candida glabrata is very critical in current therapy, and there are up to 20% reported. And it suggests that the rate of the emergence of azole resistance is much faster than the emergence of new antibiotics. So new novel treatments are needed. Therefore, in this paper, they identified the mediator and activator in relationship in Candida glabrata, and they screened for the small molecule IKIX1 and verified that the function of IKIX1 can successfully restore the azole sensitivity of drug-resistant Candida glabrata. So this is a therapeutically potential method for resensitizing the drug-resistant Candida glabrata, because it only blocks the simply protein-protein interaction, so it is also less likely to cause further resistance. So it is a promising therapeutically method. And this is my presentation. Thank you. No. In the wild type situation, they will rise. The activator will recruit the mediator only when the presence of toxic compounds such as ketoconazole and fluconazole. If there is no absence of this, there is no presence of this toxic compound, the pathway will not be activated.
Okay, and then I couldn't understand why additional IPX1 cannot further promote the cytotoxicity in response to your drugs. Because you just mentioned in the wild type situation, these two, in the presence of the drug, these two proteins interact, right?
Yeah.
So in that case, then when you add IPX1, that should also work. But
You mean in this picture?
Yes. In the animal model, I think this one it's not talking right. So I couldn't understand why.
The addition of IKX1 did not increase the survival rate
Yeah.
because it will be induced by the presence of azole. So if there is no treatment of azole,
You did add azole.
yeah. Azole is this group.
Oh, okay.
Azole. No. The azole is this one, the blue line, and the yellow line is with IKX1.
Plus azole.
Yes.
Yeah, so I would suppose that with the additional IPX1 should further improve the survival rate.
Because the entire pathway is induced by. Like the author said that in the wild type, the addition of IKX1 will not increase the survival rate. But in this picture, it shows that, and in this picture, it shows that the addition of fluconazole of IKX1 with the azole can, in a small amount, can decrease the fungal burden and increase the survival rate. So the author suggested that the addition of fluconazole and IKX1 can also be used to the wild type strains, but it does not reach a statistically significant level. Yeah.
现在有同学要提问吗？那我们就谢谢陈润同学。